Malaria Research Laboratories, College of Medicine, University of Ibadan. From 2000 to 2003, as has been mentioned, he was postdoctoral research fellow, Department of Immunology and Infectious Diseases, Harvard School of Public Health. From 2001 to 2005, he was a consultant molecular biologist for the World Health Organization's anti-malaria resistant drug network in Africa. From 2000 to 2013, he was a visiting research scientist, Division of Experimental Therapeutics, Walter Reed Army Institute of Research, Silver Spring, Maryland, also in the United States of America. In 2006, he was a visiting faculty, Department of Preventive Medicine, Faculty of Medicine, Federal University of Rio de Janeiro, Brazil. From 2009 to present, he is a visiting professor, Harvard University, Department of Immunology and Infectious Diseases, Harvard T.H. Chan School of Public Health. From 2011 to present, he is Professor of Molecular Biology and Genomics, Department of Biological Sciences, College of Natural Sciences, Redeemers University, Ede in Osun State, Nigeria. Professor he has published extensively in the domain of immunology and infectious diseases. He has served and continued to serve on the editorial board of many peer-reviewed journals. His research findings has been presented at workshops, seminars, and conferences. Distinguished guests, ladies and gentlemen, may I present to you a distinguished academic, an engaged and intensely focused researcher, Professor of Molecular Biology and Genomics, and Director, World Bank-funded Africa Center of Excellence for Genomics and Infectious Diseases, Redeemers University, Professor Christian Happy.
Thank all of you for being present at this meeting, this workshop. I also want to use the opportunity to thank the management of Delta University to facilitate my coming down here and share with you some of the knowledge that we have. I want to use this opportunity to say that it is a unique and great pleasure to have to be invited here by Professor Patton. It took him over 2 years to get me here this morning and for these few hours. Not that I want to come here, but it's just that the schedule is just too hard, too tight. And I find it very difficult to move around because of my international, schedule. I know this workshop would have held 2 weeks ago, but I had to ask for it to be canceled because I have some other commitments. And I really want to say that I'm pleased to see the progress that is done in his career. Unfortunately, I wasn't here to honor him for his inaugural lecture, but I was very pleased to see that he became a professor and gave an inaugural lecture in this prestigious university. Professor, I remember years back, during his PhD program, he learned his first steps of molecular biology in my lab and worked with us, and I'm so pleased and proud of you for all the things that you've done. In master's and in his PhD, and then eventually we are our colleagues, we are our professors together. That's what you want to see. And I really want to commend him from all the progress. I mean, he's someone I'm very proud of. Before I get started, so I really just want to use the opportunity also to tell them, and I'm very pleased to see a university that has so much potential, and there's so much to be done here. But let me use the opportunity also to announce, and I told him yesterday, that having seen what is on the ground, the doors of my labs are now open for students of the Niger Delta University. So, and as such, we believe that we want to work together and then set our foundation that can actually enhance what you have here. You started with a small lab, but I'm sure that lab will continue to evolve. But we also want to make serious investment in human resource development in order to boost your lab and then bring your lab to the level where the world effectively will respect what you're doing here. Thank you. Now, let me get this topic. Professor Taffern really surprised me because when he called me, he knows the time is so hard, but for the past 2 years it's been bothering me. And at some point this year he said, well, you got to do it. Do me this favor. I said, well, I'll find time and come. Eventually we were a lot of back and forth. He obliged me to be here. And he asked me, do you have a specific topic in mind? I said, I don't know, I have no topic. Whatever you ask me to do, I'll do it. But I was very surprised when he sent this topic to me, unraveling the mystery behind DNA and RNA. Then I told myself, how does this guy want me to start unraveling the mystery behind DNA and RNA? Was he told that I'm what the Yoruba call Babalao? I'm not a witch. I'm not a witch. I'm not a wizard. How do you want me? The witch and wizard are the ones that I see beyond. I'm just a mere human, so I see the things that I hear. But I'm going to try, and I started thinking, I said maybe I have a potential to see beyond what we all see. Then I said, okay, let me try and see how I can do justice on this matter. But thinking about it, I really realized that there's so much to be told, to be talked about. But I said, let me get started and then see what really DNA and RNA are all about. Well, if you want to unravel the mystery behind it, then it means that you need to go back to the origin of life. Because the DNA and RNA, as you see them as molecules, they are actually the foundational or the fundamental, the foundation molecule of life. They are the molecules that are responsible for life. For anything that lives, there is a DNA or RNA in there. If you take out that DNA and RNA, then life stops. Then I started ruminating about it. And then I started thinking about it. And then what does that mean? So it means that for God, when he was creating the world, he definitely created the DNA. And effectively, when you hear and listen to the Bible, you realize that, or you read the Bible, you realize that actually God used the DNA as a molecule to put life into man. Because when you read it, you will see that God created everything. When he wanted to create man, he created man from mud. But then the Bible will tell you that he breathed life into man. When you hear about breathing life, what is life? The fundamental unit of life is DNA or RNA. So which means that at that moment, because you are human, we can understand it, God actually inserted DNA into man and then man started living. That's what that means. As a scientist, when you read and then look at the Bible, you realize that this is exactly what that means. That is breathing life. We can only, as scientists and people, see breathing. But then, because it is proven, for anything that is living, if you take out the DNA or RNA into that thing, then life stops. So it means clearly that for life to go on, then there is that molecule that is crucial. So without DNA or without RNA, there's no life. So for man to live, and for God to give life to man, he had to breathe that thing, which was an insertion of DNA into life. And then, I start thinking further, and then you realize that, when we talk about the mystery behind it. It means that the fundamental unit of life is the DNA. But then when you want to see, when you talk about diversity, which means that the DNA is responsible for anything that we see. If you change the DNA, then something will change in a person. And that's what God did again for the second, what I call the most important experiment that God did in the world. We all hear about creation. God is the same engineer. He's a molecular biologist. He's a biotechnologist. God did all of that. He's the person that did the first cloning experiment. But we know we are so short-sighted. It took us millions of years to research and understand the nature of God. And that's what we call it research, right? So which means that if you are researching, it means that somebody searched before you. You are just researching. So God searched and realized, oh, we need to do something. Next slide. And that takes me again to Genesis. So that gives you the structure. I don't want to dwell onto this because I know you all know what DNA and RNA are. Double-stranded DNA molecule for DNA, and is a single-stranded molecule. The RNA is actually a copy of one strand of the DNA. But then there are some few differences, right? So those few differences are just in the nucleotide. So here you see, for instance, the nucleotide that go for the, that are used to encode an RNA molecule. On that end, you see those that are used for the DNA. But then you realize that, they are interwoven and interlinked. They depend highly on one another. So you can have an RNA without a DNA. And from the RNA, then you can have proteins. And those proteins are basically just translating the message that are into the DNA. So which means that we are the reflection of our DNA. And as such, when God wanted to make changes, He created man. Next slide. And then if you go into Bible, you go to Genesis 2 verse 21 to 23, this is what it tells you. That when God created man, then the Lord made the man fall into a deep sleep. And while he was sleeping, he took one of his man's ribs and closed up his flesh. He formed a woman out of the rib and brought her to him. And the man said, at last, one of my own kind, bone taken from my own bone, and flesh from my flesh. Woman is her name, because she is taken out of man. But then, ask yourself the question, why did God do that? And how did He do it? If you read this very well, you will realize that when you put someone to sleep, what you call today in medicine anesthesia, right? That's what God did at His own time. When you put somebody to sleep, he's in between life and death. That's what you do during surgery. But then God took a bone. For Him to take a bone, He had to open up. So He did a surgery. And that's laser surgery. It's only with laser surgery that you don't see the marks. So again, it tells you how God is just so powerful. We call ourselves all of these things, but He's done all of those things. He did a surgery and laser surgery. And then from that surgery, He took something from the man and then modified that thing. The thing that He modified to create a woman, when you do that in genetics, in genomics, in molecular biology, we call that gene cloning. So God, millions of years ago, cloned woman out of man. So these are the mysteries. We are unraveling this mystery behind the DNA. So He created woman out of man. And He made modification, and then those are the reasons why you see those changes, those differences between women and men. But then you ask yourself the simple question, why did God have to create a woman out of man? Because remember, God created man and He said, you are going to be governing over my whole world. You'll be in charge. But it's obvious also that when God created man, He was there, He was observing this guy in the Garden of Eden. He realized that guy had violent and dangerous tendencies. He had destructive tendencies. He realized that if he let that guy alone in the world, he was going to destroy the world that he created. And he had to create a moderator.
He had to create a moderator. And that moderator, he actually did it so well. He created someone that is more beautiful. He created someone that is stronger. And he created someone that is more intelligent. Those are the mysteries we are unraveling. That is the thematic. We are unraveling. And to confirm his experiment, you see, God doesn't work just like that. He created this person and he wanted to test the success of his experiment. What he did first was to do the first test of intelligence. He created man. He told man and gave him all the instruction in the garden. Do anything, but then, when it comes to this fruit, don't go near. But to test the person to know that this lady that I created will be able to control this guy and maintain the world that I created. He first of all wanted to test the intelligence. But you know what? Eve came, despite all the instruction that God gave Adam, she was able to convince him to go and do what God did not ask him to do. That shows someone that is more intelligent. And I can tell you, it runs up to today. We're all men and women here. Women are very manipulative. If you want something, whether you like it or not, one way or the other, you will give it. We all know that. If you want something, she knows she likes food, she will get that food that you like most. And while you are eating, she's singing something in your ear. Before you see it, you will say, oh, you know that my soup that in the thing. There's a check there. Go and get it. It's intelligence. They're very intelligent. So that was the first test. God confirmed. Because it's very difficult. Imagine you are with your dad in a room. Your father tells you that never enter this room. For some lady to come and tell you that don't listen to that man. Let's go in there, let me see what is in there. She must be very intelligent, very manipulative to break, that instruction. That was confirmation of test of intelligence. Now come to the test of strength. God created a stronger human being, which is a woman. They are emotionally very strong, and they are physically very strong. Physically, I say. And to testify this, God says, go and multiply and fill the world. Who is supposed to be responsible for bringing the children to the world? Why didn't God trust the man to do that? Because God realized that this guy is weak. He couldn't just cope. The guy is very weak. And to confirm that, listen, if you go to your home, and then a man has a little wound, a little cut somewhere. When a man has a cut in his finger, he goes around with his finger like this. Oh, my finger is killing me. My finger is killing me. But look, he's not giving. But when a woman goes to the labor ward, you don't hear about it. She will do that. Another 2 years she'll be there, or even next 1 year is there. But I can tell you, if the men were to fill the world, the human race would have been extincted. Because men do not. They don't cope with pain. We don't cope with pain. We hate pain. We don't want to hear anything. And I can tell you, if men were to want to give birth, look, once you go there once, a man will tell you, never ever in my life. That's the truth. That was a test of strength. It gives you, and it tells you that he created someone that was more intelligent, someone that was stronger, and then someone that is more, that is more intelligent, strategic. If you go back in the histories, be it in Europe, be it in Africa, be it anywhere, who were the winners of the wars and all the campaigns of war that were going on? We know in the history of Nigeria, for instance, that you hear about the Bini Amazons, right? They were the ones in the front, that will make the game change to change the world. The reason is that we men, we believe in so much physical force. We are brutal. We are not strategic. We don't think. We go to war, we just butcher ourselves. There's no strategy. But you realize in the end that when the women went to the war front, they were the ones designing the strategy. They were the first spies. The only reason why they were winning the wars was because the women, the Amazons, would go and cross to the other side, pretend to be gentle, get all the necessary information, and came back. and use those information to strategize and defeat the adversary. That again is intelligence. But then when you look at the genome of men and women, we all have 23 chromosomes. The men end up with chromosome Y, and the female end up with chromosome X. For those of you, I'll give you an assignment. Go and look at the length of chromosome X and look at the length of chromosome Y. Chromosome Y is a very short chromosome. Chromosome X is a longer chromosome and more dense. All of those fine, what you call refined things that you see in women, tolerance, beauty, everything, are all located in the chromosome X. So that means that's where God went to make, and He made so much difference. Again, unraveling this mystery. God created a better human being. They are better. There's no doubt. And I'm glad, to say that the future of the world is women. I spotted one lady on this ground. Please, can you stand up? I love your t-shirt. The message there is very powerful. The future is female. I was amazed to see this. I didn't know. I just saw that and I said, wow, that's the kind of message I like to see. Thank you for wearing that so that you can add value to what I was trying to say. So the future is female, and that's why God created the world. When you see, for instance, even in government, go look at countries that are led by women and look at countries that are led by male. Those led by males are the ones that are always doing wars. Wars without reason. And they are less developed. Give power to a woman. I can tell you, give a power to a woman in Nigeria. In the next 5 years, this country will make it 360 degrees. Because they are more intelligent. Because God created them so special. So, and as such, when you say we're unraveling the mystery, right? That's one of the mysteries that we just unravel. But now, over the years, what happened was, we evolved with science, and then we went from the fact of, we're still trying to understand what God did. He did so many things. We are still trying to just understand it. And we've moved from just understanding one single gene, we went to areas what we call genomics. Genomics is actually the ability to actually try to study DNA at larger scale. That is, studying the whole DNA of one person, or the whole RNA of one person. So, and over the years, as science evolved, we realized that, that new science is so highly developed in the US, is so highly developed in Europe, is so highly developed in Asia and in Africa, in South America. But unfortunately, Africa does not contribute to that, and that has created a huge divide between the way science is done and health care is given between the West and Africa. Today in Africa, because of the DNA in Europe or in the US, when you go to the hospital, they diagnose whatever you have in function of your genetic makeup. We talk about personalized medicine. We don't do, the way medicine is done in that part of the world is no longer the way we do it here. When you go to the hospital, they don't just prescribe just any drug. They prescribe the drugs based on your genetic makeup. That is basically customized diagnostic, customized treatment, customized vaccine. And that's exactly what the DNA science and the RNA science is done. And the same way when they research, they are researching based on DNA and RNA. Unfortunately, Africa is not in the game. So this is one of the reasons why I decided actually to move the game back to Africa, because I realized over the years, while I was at Harvard, I was educating those that have the resources and those that have a lot of knowledge already. Then I just said, okay, well, maybe it is time to come back and then try to see how you can reduce the knowledge gap and then the divide that is existing within Africa and then the rest of the world. And that's the reason why, for instance, we created this center called the African Center of Excellence for Genomics of Infectious Diseases. But then remember that genomics is all studies of DNA and RNA. And that's why, we'll see over the course of my talk how we unravel DNA and RNA to study problems that are peculiar to us. One of such problems that we actually decided to study is infectious diseases, right? Infectious disease is something that is very common, and mostly to Africans. Why is it common to Africans? People say, oh, well, we have other problems. Someone told me the other day, why is it that everything that is bad comes from Africa? Ebola comes from Africa. HIV, everything that is bad comes from Africa. And I told him that, look, I don't think that they are bad things. They are actually good things, right? There's a saying that says, oh, God bless Africa with a lot of natural resources. God bless Africa with a lot of mineral resources. The truth of the matter is that God bless Africa with a lot of problems. God bless us with problems. Diseases are problems. Famines are problems. Wars are problems. These are blessings. We shouldn't look at them as problems. These are gifts. Because you have the issue of war, I see no reason why the Africans shouldn't be the champion in the world for peace and conflict management. When you do war, at least you should be able to sit around the table and solve the problem. We should be teaching the world how to do peace and conflict management. Because we have problem of infectious diseases, we should be the one teaching the world how to solve problems related to HIV, malaria, and everything. We are rich with viruses. We are rich with parasites. This is well. This is well, but we are not looking at this as well. We look at it as a problem. We should convert those things into, work. Convert this into work, because we should be able to bring foreign exchange based on the fact that we are teaching the world how to solve the issue of all these problems. There's no reason why Africa should be the one teaching people how to do, good agriculture. We have problems, but we should be solving them. We shouldn't be looking at them as problems, they are challenges. But use them as stepping stones to address those issues. So, and that is based on that kind of, attitude that we created this center. And this center, as you saw in the previous slide, go back. That center is a conglomerate. It is made of academic and clinical testing centers across Africa, but it's also made of but we also work with collaborative partners outside Africa, where you see Harvard, Tulane University, the Broad Institute that you mentioned, and then industry partners. But then the success, the message here is that when you do something well, you're going to be bringing more partners. So the success of this center actually brought more people to join this center. So for instance, when we started, we didn't have all of these centers. Today we have Stanford University that is started, University of Cambridge in the UK. Oxford University also joined the partnership. Then apart from that, we have states within Nigeria that are willing to do that. Lagos State came to us and they said, you know what, you save us from Ebola, we don't want that to happen anymore. So we have a memorandum of understanding with Lagos State, helping them to build the capacity and also helping them, training them how to respond if they have problems. So that has helped a lot in terms of this. But then what are our issues? Next. The mission are twofold. One is education, the other one is research. Next, education mission for the program is actually to create what we call, a vibrant academic environment that transcends national boundaries, where people can do relevant, responsive, and cutting-edge translational, research for health in Africa. Why do I use the word that transcends national boundaries? It's because I realize in Africa, knowledge in Africa is key. What you do in the Netherlands is very difficult to translate that to a movie, because there's no brain for it. There's no process. And this is something that I told some African researchers some years, 2 years ago. It is a terrible thing for Africans to start thinking about, when they talk about the African passport, they are thinking about politicians. I challenged them and I told them, if you want to move, then if you want to talk about the African passport, prepare the first African passport for academics. It's easier for me to leave Nigeria or Nigeria to go anywhere else in the world than to leave Nigeria and go to another African country. It is a terrible thing. There was knowledge, and to give knowledge to help a Kenyan institution to build. They were telling me it was going to take me 3 to 4 weeks to obtain a visa. But then if I'm leaving here to go to another European country, 2 days. So who loses? It's easier for me to go to Europe and share knowledge than to go to another African country. So, and it's a shame that we don't have all these barriers that are preventing what I call brain mobility. But then, generally, the plan is actually to build a critical mass of white African trained scientists that will use knowledge of genomics to address issues related to infectious diseases. Those are the aims. But in order to do that, you need to create, there's really no curriculum for genomics, for genomics, to teach genomics in Africa, which is studies of DNA and RNA. So what we did was actually to set up a curriculum, and then we set up a master's and a PhD program at the university, and then within 2 years, these two programs went from no accreditation to have a national accreditation, and effectively have an international accreditation. So now we have a master's PhD program that is equivalent to a master's PhD in Oxford, University of the European Union accreditation. It's poor. It means it is specific. It means that if you set up mind and goals and things, and we can do it. Right? But apart from doing that, we also engage in community. But then the research component has to do with something that is very peculiar to us. Next slide. It is addressing issues related to fever. You know that, in everywhere in Africa, fever, right? When you suffer from anything, you start with fever. But unfortunately, in our context, once you have fever, the first culprit often is what? Malaria. And malaria is so misdiagnosed and so mistreated. Because once you have fever, you think it's malaria. But when that malaria fever is not treated, then the doctor will tell you it's typhoid. And then we'll evolve, we'll keep graduating. We'll graduate to a level where our doctors will tell you you're trying. And that's why we decided to do a research that is going to be addressing the issue of fever. And then the fever is actually to identify across the whole of West Africa, the whole of the West African region, what are the pathogens that are driving fevers, so that we can develop what we call a one-stop shop diagnosis model that will address all the fevers in a given environment. So, but then ultimately, our goal is to create what we call the next generation of African pathogen hunters. We always play what we call defense. The Vice Chancellor was referring to Ebola, Lassa, monkeypox in the Niger Delta. But what happened? Do we have the ability to diagnose them before they come back to hunt us? No. We're always playing defense. We play defense. Once there is a monkeypox outbreak, you see all of us are running helter-skelter. Tomorrow, Lassa fever, everybody start running around. Do we have the ability to identify them and characterize them before they come to us? Yes, that is the answer. Can we do it? Yes. Can we do it better than those Caucasians? Yes. Do we have the resources to do it? Yes. The only problem is that our government misplaced their priorities. Instead of putting the money where their mouth is, they go and put money elsewhere. And the challenge I always say is, look, the Niger Delta alone can decide to be a hub to address all the problems of Nigeria, but provided that the government in Niger Delta set that as a priority. There's really no point to go and build a house in Dubai and buy things all over the world and then go to the US and everything. The reality is you're going to come back here. The reality is that you will die back here. The reality is that in the end, having saved your money all the Swiss bank, they will tell you that they don't give you visa, then your money is locked there. The reality is that we will end up dying in the same hospital that we neglected. So do we have the power and the energy to do this thing? Yes, we can. Next slide. Now we'll talk a little bit how you use genomics to achieve a few things. And this is basically just a case study. And the whole reason why I talk about this is actually to let you know that you are so powerful. That God created you so powerful. And that if I was able to do it, then it means that you can do better. Remember I told you what? I am a man. I can tell you if the lady decides to do what I'm doing, they will do it better. And then much more, and then they will do it better than myself. So I throw the challenge everywhere I go to the young ladies. Please, go into science. Let nobody tell you you can't do it. Those men telling you that you cannot do it is because they are afraid. You do it better than them. Next. Now, if you remember, in 2013. Ebola did not just start randomly. It started in some small villages in Guinea. And in Guinea, the world was looking away. Nobody bothered about it. I had a premonition that that disease was going to spread and kill all of us. What I thought of doing was to think, oh, well, let me gather some friends and then we'll see how we can address the issue of Ebola. I was kind of ambitious, you know. I thought, you know, once you have a good brain, then you can solve all the problems. You don't realize that even with a good brain, you also need resources, and then you also need to play politics. Then you see on that picture, I have some friends here who I met, and then we identified one of the key issues was diagnosis. Even if Ebola came at that time, that was toward November 2013, how would he know that it's Ebola? So we realized diagnosis was something that was very important. We needed to put in place diagnostic, platform. So what we did was basically to, first of all, go to the database, identify diagnosis. The diagnoses were not very good, but we actually developed new diagnoses and then trained people on how to diagnose Ebola. We did the training in Nigeria, we did the training in Senegal, we did the training in Sierra Leone where we were working before. And as I rightly, thought about it, on May 20th, the first case of Ebola was diagnosed in Sierra Leone, and it was diagnosed by one of the guys that I trained, Augustine Gober here, to diagnose Ebola. But what was interesting was the fact that, yes, he diagnosed, but then we needed to start knowing how the disease was spreading. So we activated what we call the contact tracing mechanism. Within the first 7 days of the outbreak, we had 35 suspected cases, confirmed 14, and we were able to bring down, through contact tracing, bring down the origin of Ebola in Sierra Leone to a traditional healer. This traditional healer thought that he had all the power. He was busy treating Ebola patients, thinking that he has all the powers to treat and to manage patients. Yes, eventually he pretended he could manage and eventually died in the process. But then, what was most important for us is that when we had it, we started thinking about how do we solve the problem? How do we prevent this from spreading? We went on and then started thinking about using genomics as a way to sequence. Because one thing is, basically, once you have diagnosed, then what do you do with the information? So we thought about using genomics as a way to make the information available to the world, where they could use people that are developing bad diagnosis could develop better diagnosis. Those that are developing treatment could develop better treatment. And then those that are developing, vaccine could do better vaccine based on the viral sequences. And what we did within the first 3 months of the outbreak in Sierra Leone was basically to sequence about 99 viruses, and we made them available, open access. That is, make it available to the world to use. But more importantly, because we were aware and then we were prepared, we actually averted what we call in the world what could have been one of the major health calamity and disaster in the world, by diagnosing the case in Nigeria within hours of being alerted. I got a call around 8:40 in the night on January 22nd that there is somebody that there's a sample we suspected that it could be that we tested malaria, but this malaria case for this man is not going away. But then he's Liberian, so we don't know. So I had to leave my house in Ibadan and travel on Lagos-Ibadan expressway. I don't know if some of you have gone through that road. It's one of those most dangerous roads in Nigeria. What was driving me, I didn't know. I told my wife I was going without my driver, but driving myself at that late hour in the night. But I still drove. And I still went back to that lab. And I still walked all night. Then by 6:40 in the morning, I was able to call not only the hospital to I told Dr. Badevo that guy has Ebola. Under no circumstance does he go out of that place. I understand later on that it was in that process after I told her that she was fighting with the man that wanted to leave. And that's probably how she got infected. And that's how forever she remained my hero, because she died, but she saved millions of lives in Nigeria. If not of that heroic action, were we able to do the diagnosis within hours and provide information, this gentleman would have continued his trip to the southern part of Nigeria because they were coming to Calabar. He would have probably along the line kept infecting people, and we would have had a situation whereby in Nigeria by what we call by extrapolation, we would have lost millions of people in this country. Because if in Sierra Leone with small 4 million people they lost thousands, imagine a country like Nigeria with 200 million people. You extrapolate, then it becomes worse. So I'm sure we would have been talking a different story today. Would have actually, many of us wouldn't have been alive. But then it shows the power behind the technology and how you can use genomics, and they will talk about unraveling the mysteries. This is how you use the DNA and the mystery behind the DNA, understanding RNA, to now solve problems. So again, this is process of unraveling DNA and RNA mysteries. Next. What we did was basically, as I said, sequence 99 genomes, made them available, open access, anybody could have access to the genomes. But then we also realized that at that time, because of panic, many people used to go, once you have a fever, in afflicted countries, it was stopped. It was Ebola, right? People with malaria, as you see there, will go to the Ebola treatment center. They had only malaria, but unfortunately they go to the Ebola treatment center, they get in contact with the Ebola patient, and then they come back to their homes infected with Ebola. So, and that we have to devise ways of using one single diagnosis to do differential, and that's what we're doing in one single assay to do all of these pathogens. And you can see, for instance, these are some of the pathogens like Plasmodium, malaria, HIV, Lassa. So this is what we could do in order to say, well, we need to resolve this issue. And tell people right there almost every time that this is not working, that is not Ebola, stay home, right? But then in addition to that, we went on to develop new methods of sequencing, that people can use all over the world to to better understand the genomes of the Ebola virus disease. Next, we set up a system that I will call up to today is a reference, which is unprecedented. An assay that would take us a month to do, we're doing that in 10 days. So you can see here, from sample collection to releasing the data on NCBI, it took us 10 days. Up till now, I tell myself, where do we get the energy? How do we make it? Because we were doing that during 10 days during the Ebola outbreak. But in my lab, we can't even do that in 10 days anymore.